Hello, everyone. This is Chapter 9 from Essential Biochemistry, and I'm covering sections 9.2 and 9.3. So we want to start out by talking about membrane transport, and we can sort of group these into maybe two categories: non-mediated transport, which is just simple diffusion. So this is diffusion where a substance could go across the membrane and there's no protein transporter required. So generally lipid-soluble molecules of any size can cross the membrane because you might remember from chapter 8 that we have lots of fatty acyl chains in the interior of the membrane, which are quite nonpolar. So sort of in the like dissolves like uh, 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 idea that nonpolar substances can go across the membrane without the need of a protein transporter. But these other types right here do need a protein transporter. So these would be water-soluble substances that wouldn't be able to go across the membrane on their own. So we're going to look at channel-mediated, which is passive transport. So here this shows a yellow molecule going through a channel and generally not interacting with its transporter. Transporter-mediated, which has a specific binding site for the bound substance that will be transported. So this is shown in yellow and in red, and here we can see that we're going along a concentration gradient where the red and the yellow molecules are going from an area of high concentration to low concentration. That is contrasted with active transport. So if we have this blue square right here, if we're transporting it from the inside to the outside, at least the way it's drawn here, you're going against a concentration gradient and the input of energy would be required. So let's look at examples of all of these. So first of all, though, before we dive into the different membrane proteins, we should note that membrane proteins carry out various functions in association with the membrane, and they interact favorably with the membrane of necessity. So we can classify these as three types: integral, peripheral, and lipid-linked. So integral membrane proteins completely span the bilayer. Peripheral membranes will be associated with either one side of the leaflet or the other. And a lipid-linked protein is anchored to the protein by a fatty acyl chain that interacts favorably with the fatty acyl chains of the phospholipids that make up the membrane. So let's look at this integral membrane protein. They are asymmetric. So the side of the protein that faces the outside of the cell will be different than the one in the inside of the cell. If you want to dissociate these proteins from the membranes, you can, but it can be difficult because you'll need detergents. And detergents will help strip the membrane away from the protein, but the fatty acyl chains of the detergent will interact favorably with the hydrophobic portions of the protein and make it soluble. Because as you can see here, it makes sense that these domains of the protein that are in contact with the fatty acyl chains have to be hydrophobic amino acids. So this is shown here where the artist has drawn the the amino acid side chains that are hydrophobic in green. And the side chains extending into the aqueous environment, whether you're in the outside or the inside of the cell, are more hydrophilic and are shown in purple. So we can see that generally that the region spanning the membrane consists of non-polar amino acids, whereas once you get to those polar phospholipid head groups and the aqueous media on either side, you'll want to have polar amino acids that interact favorably with water. Here's an example. This is glycophorin A. It's found in the red blood cell membrane, and it spans the bilayer once. It forms an alpha helix, actually, which is not shown here. But we can see that there is a big difference between what we see on the inside and the outside of the cell and the part of the protein that crosses the membrane. So we see lots of polar amino acids interacting with the aqueous environment outside the cell as well as inside. We can also see that this protein is glycosylated, which is shown here. The specific linkages are shown here. These little diamonds indicate that the binding of the bonding of the sugars to the specific amino acids. And again, you notice that the sugars are attached to the domain of the protein that faces the outside of the cell and not on the inside, and that has to do with how they're synthesized and how they get to the plasma membrane. If you look at the amino acids that are crossing the membrane, you can see that generally we see the hydrophobic amino acids, isoleucine, leucine, phenylalanine, valine, methionine, alanine, glycine, isoleucine. We do see a few glycines right here that can that are smaller and are not either significantly polar or nonpolar. So the amino acids that are generally in contact with the fatty acyl chains will be the nonpolar amino acids. And you can see there's a switch over from nonpolar to polar right here when you go from an isoleucine to a serine and a tyrosine over here, and from an isoleucine to a glutamate over here when the protein is now interacting with the aqueous environment on either side of the membrane. Here's another example. This is bacteria rhodopsin. It's a bacterial protein. Scientists are interested in it because it looks a lot like rhodopsin, the protein in our eyes that is responsible for vision. This is a 7-membrane spanning alpha helix where you're essentially going back and forth across the membrane 7 times. And again, the alpha helices, the amino acids that make them up, that are in contact with the fatty acyl chains will be nonpolar, whereas the portions of the helices exposed to the aqueous environment will be composed of polar amino acids. Here's an integral membrane protein, AMP-X, I guess. I'm not sure how you pronounce that. It's not something that I work with, so I might not be pronouncing it correctly, so I apologize if that's the case. These are hydrogen bonded beta sheets, so this forms a beta barrel, and that forms the structure of the transporter. And again, you can see that the hydrophobic chains, which are green, are facing the bilayer, and even without having the bilayer here, you can, if you look at the orange, which are these aromatic groups, tryptophan and tyrosine, you can see that they're located mainly near the lipid head groups, because tryptophan and tyrosine have non-polar portions as part of their amino acid side chain structure, but they have polar bits as well. With tryptophan has the phenyl group, and the indole group has an NH group. So you can see where the boundary is between the portions of the protein that are interacting with the fatty acyl chains in the membrane, and those amino acids that are interacting with the phospholipid head group. Sorry. Here's another diagram right here that shows the various kinds of lipid-linked proteins. So here, proteins are covalently attached to a fatty acyl chain, and this anchors them into the membrane. Proteins labeled 5 and 6 right here are lipid-linked proteins. Proteins labeled 7 and 8 are peripheral proteins. They are just sort of loosely associated with these integral membrane proteins right here. So these proteins are actually fairly easy to dissociate from the membrane if you want to study them. Usually either a salt solution or pH change will be all you would need to dissociate these proteins from the membrane. So let's go back to our list of mediated transporters. Channel-mediated transport is described as passive transport. So these substances are going along a concentration gradient. They can be pores or channels. So pores are always open. So bacterial porins are not very specific. They allow the passage of small polar molecules. Channels open and close in response to signals. So ion channels and aquaporins will be able to open and close. So again, this is an example of a passive transport. So let's look at some examples. So we can first look at channel mediated. So pores and channels provide passage for small molecules and ions. So here, the example in this diagram is passive transport, where we're going along a concentration gradient. And in general, the transported solute doesn't interact with the protein transporter. So here's another sort of I guess a cartoony kind of picture that shows these red small solute molecules going across a pore or a channel and going along the concentration gradient. So here are some examples. So the AMP-F porin that I just showed you is channel mediated. So we usually use the term pore in bacteria, but you can see that these beta barrels that I that you see right here allow these are water-filled pores. So this allows the passage of the polar substances across the membrane. And this diagram, I think, shows this pretty well because the artist here has just shown the polypeptide backbone. So you can see that there's space for small molecules to go across. Here's an example of channel-mediated transport. Now, there is an interaction here that is important. So this is a potassium ion channel, and this is channel-mediated transport, but this channel is highly selective for potassium ions. And the way that this works is that over here, this is the purple potassium ion, and there are actually some favorable interactions that form between the positively charged potassium ion and the carbonyl groups of the peptide bonds. And this explains why this channel transports potassium and not sodium. The sodium ion is a lot smaller and it wouldn't be able to form those favorable interactions. So this is highly selective for potassium transport. So let's look at another example, aquaporins. So these are in mammalian tissues, and they are expressed in tissues that transport water. In glaucoma, the aquaporin 1 transports too much water, leading to pressure in the eyes. Aquaporin 4 can be a problem in the brain after a head trauma because it allows water transport, leading to brain swelling that can potentially be fatal. But you'd expect to find, under normal circumstances, aquaporins in tissues that transport water. The way that this works is that water molecules need to shed their associated waters of hydration. You should remember from chapter 2 that water molecules are highly hydrogen bonded. So you need to pass one water molecule at a time. The other thing is that we can't be transporting hydronium ions or hydroxide ions because this would change the membrane potential because they are charged, and also potentially the pH. So this is what the aquaporin structure looks like. So there are a number of pictures right here. So what's interesting is that this protein traverses the membrane via these alpha helices. This is kind of interesting right here in that this kind of looks like a single alpha helix, but it's actually different domains of the protein. This picture right here, these red dots are meant to represent the water molecules. And here they're just showing the oxygen of the H2O and not showing the hydrogens. And here's another example right here from our textbook of the aquaporin. So here's an example of how this works. If we want to transport water across using the aquaporin, you notice we have our water molecules here. Here's an arginine right here that forms a hydrogen bond with this water molecule right here, which then is passed along to a histidine, and we get a narrowing of the channel right here that allows a single water molecule to pass, and this is the asparagine right here. The next thing that will happen is you can see this water molecule is kind of sort of flipped kind of sideways right here, so that you have a single water molecule oriented very specifically, forming hydrogen bonds with these two asparagine amino acid side chains. So here we're orienting the dipole very specifically, and you can see that the way that this works, you'd only be able to transport one water molecule at a time, and you wouldn't be able to form these interactions with either hydroxide or hydronium ions. And then the water molecule is passed along to the rest of the channel. So these amino acid side chains, these polar amino acid side chains that face the inside of the aquaporin, specifically orient the water molecule to transport one water molecule at a time, and to prevent the transport of hydroxide and hydronium ions. Peter Agri and Roderick McKinnon won the Nobel Prize for determining the structure of aquaporins, because before aquaporins were discovered, it was known that water could cross membranes, and water is polar. And the idea was that water would pass by just by simple diffusion, just because there was so much of it. So their discovery of transport proteins that accomplish this process in a very specific way was groundbreaking. Okay, so let's go to transported mediated passive transport. Sometimes this is called facilitated diffusion, and here there are specific binding sites for the transported substance. And again, if we're in passive transport, we're transporting along a concentration gradient. So here are two diagrams from two different artists. So this is just a general diagram right here where we have these red molecules and one binds to a specific binding site, and there's a conformational change that in just a still figure, it looks as if the transporter is flipping in the membrane. That's not what happens. There's just a conformational change. Imagine that these two subunits are brought close together, and then these two subunits come apart to allow the transport of this substance. This shows a glucose transporter where we have glucose shown as a hexagon binding to its transporter. And again, you have a conformational change that brings these domains together, separates these two, and glucose dissociates from the transporter. And then there's a conformational change that reverts the transporter to its original conformation. So again, in still figures, it could look like there's that somehow this transporter is flipping in the membrane. Then that's not what happens. So here's another picture right here that shows a little bit more specifically in chemical detail. This is GLUT1. So GLUT is glucose transporter. There's a whole family of glucose transporters, lots of different kinds of glucose transporters in different cells. And this shows glucose in its chair conformation, binding to its binding site, and then the conformational change that allows the glucose to be released on the other side. So I have a very short animation that shows this, and I like the animation because I think that this shows pretty well that there's not a flipping of the transporter in the membrane. So let's just, okay, so this is on YouTube. Hopefully, okay, I'm not going to make a see an ad. All right, so here we have glucose up here, and the glucose is going to bind to its transporter. So there's a very specific binding that occurs, and now glucose is bound to its transporter. And then there will be a conformational change that was just shown just right there, and then that allows the glucose to be released on the other side. So I think this shows pretty clearly the conformational change that occurs. All right, so let's get out of there and go back to our PowerPoint. What you can do is you can go into the lab and you can measure glucose transport, and you get this familiar hyperbolic curve. So this curve is consistent with the model of glucose transport shown in the previous slide, where we can use what we've learned about Michaelis-Menten kinetics and calculate Vmax, which looks like it might be, I don't know, maybe about 0.8 millimolar centimeters per second times 10 to the 6th. We can calculate a Km, maybe that's about 0.4, drop a line over, drop a line down, we get about maybe 0.5 millimolar for our Km, and that Km could be considered to be an affinity constant. Or at the very least, it's the concentration of glucose you need for the transporter to be half maximally occupied. Notice when the glucose concentration becomes very high, the glucose velocity does not increase because the transporters become saturated. So that's just like the enzymes becoming saturated with substrate at high substrate concentration. The hyperbolic shape is consistent with the model of one glucose binding to the transporter at a time and a single binding site in which there's no cooperativity. So had there been a transporter involving multiple binding sites with cooperativity, you would see a sigmoidal shaped curve. But here we see a hyperbolic curve, so that is indicative of a single binding site. So I just want to introduce these terms: uniport, symport, and antiport. This occurs with substances that bind to their transporters, which this diagram doesn't show very well. But it does show that uniport involves the transfer of a substance from one side of the membrane to the other in just one direction. Symport allows the transport of two substances in the same direction, and antiport allows the transport of two substances in the opposite direction. So let us go to our last category, active transport. Here we are going to transport our little blue square against its concentration gradient. This is going to require the input of energy, and we will use the energy of ATP hydrolysis to drive conformational changes. So let's see how that works. Well, first we should note that cells generally maintain a low concentration of sodium ions inside the cell, between 5 and 15 millimolar. Outside, the concentration is 145 millimolar. That means that sodium ions can leak into the cell pretty easily, which means that the ATPase needs to kick them out. The potassium ion concentration, in contrast, is kept inside the cell at about 140 millimolar, and outside it's 5 millimolar. So what the sodium potassium ATPase does is that it ejects sodium and brings potassium in. And because both of these ions are being transported against their concentration gradients, we're going to require energy in the form of ATP hydrolysis. So here is a diagram of what the sodium potassium pump looks like. It's interesting that there are these cardiotonic steroid binding sites. Wabane is an inhibitor of the enzyme. So if wabane binds to the sodium potassium ATPase, it cannot function. And that means if sodium leaks in and brings water molecules with it, and the pump cannot function to eject those sodium ions, then cells will treated with this wabane will eventually swell and lyse. So that's shown over here. So this shows the extracellular space and the cytoplasm right here. So this gives a more maybe more detailed chemical picture of what the pump looks like. So although here it's just showing the alpha subunits, we're not showing the beta subunits. In orange right here, we'll see in a minute that we're going to phosphorylate the pump in order to drive a conformational change. So we'll phosphorylate the pump right here. These two binding sites are shown. Those are binding sites for 2 potassium ions. So we'll see in a minute that we're going to be transporting 2 potassium ions. So this is how it works. So if we look up here, the pump will bring 2 potassiums in, and it will eject 3 potassiums out. ATP will be hydrolyzed, and this maintains the concentration of sodium inside the cell low relative to the outside, and a high potassium concentration relative to the outside. So if we look at the mechanism here, we can see we have a conformation, we'll just call this E1, and this conformation has 3 high affinity sodium binding sites, and there are low affinity potassium binding sites. So the 3 sodium ions bind, and what happens is the pump is phosphorylated. So ATP is hydrolyzed to ADP and phosphate, but the phosphate isn't released. It is actually covalently attached to an aspartate. So that's shown over here. So the aspartate on this E1 is phosphorylated, and the placing this phosphate group on the aspartate drives a conformational change. So we call this E2. And what that does is that exposes the sodium ions to the extracellular portion of the cell. And these are low affinity sodium binding sites, so the sodium will be will leave the transporter. This also exposes high affinity potassium binding sites. So if we go back down here now, the sodium ions dissociate because they're now bound to low affinity sites, and the potassium ions are now these are high affinity sites, so 2 potassium ions bind, and then that drives the hydrolysis of this aspartyl phosphate. So now we're just right back to aspartate, and we release the phosphate into the cell, and that drives the conformation from E2 back to E1. So now these potassium ions, these are low affinity binding sites for the potassium ions, and now we have our high affinity sodium ion binding sites exposed, and now the whole cycle can start all over again. So this is the same thing is shown in our book. I think instead of going through this again, though, because I prefer the figure that I just showed you, I'm going to show you a video. They will not show the stoichiometry, but it does show the conformational changes that occur when the pump is phosphorylated and dephosphorylated. Okay, so let's wait for YouTube to load this. It is taking a while for some reason. Okay, here we go.
In the cytosol, the sodium ion concentration is kept low relative to the extracellular fluid, and conversely, the potassium ion concentration in the cytosol is kept high. Like water behind a dam, these gradients harbor potential energy that the cell taps to fuel cellular work. Animal cells use a membrane pump, called the sodium-potassium pump, to maintain these ion gradients. To begin the pumping cycle, sodium ions enter binding sites on the cytosolic side of the pump. Although there are 3 sodium binding sites on this pump, for simplicity, only one is illustrated here. Pumping sodium against its concentration gradient requires energy, which is provided by cleaving ATP. ATP transfers a phosphate group to the pump in a high-energy linkage. Phosphorylation causes a dramatic change in the pump's conformation, so that the sodium ions become exposed and released outside of the cell. This action also exposes binding sites for potassium ions in the pump. Although there are 2 potassium binding sites, for simplicity only one is shown here. Binding of the potassium ions triggers release of the phosphate group and the return of the pump to its initial conformation. The potassium is then released inside the cell, and the cycle repeats. A complete cycle takes about 10 milliseconds.
So again, remember that we have 3 sodium ions, not just one. The pump is phosphorylated. That drives the conformational change to E2. We have low affinity binding sites, so the sodium is released. This exposes high affinity binding sites for potassium. That triggers the removal of the phosphate by hydrolysis. We drive the configuration back to the E1 conformation of the pump, and the cycle starts all over again. All right, so I do wish they had shown the stoichiometry, but anyway. So, in conclusion, we have talked about all these different kinds of transport. Non-mediated transport just involves a nonpolar substance going right across the membrane because it is able to interact favorably with the fatty acyl chains of the phospholipids that compose the membrane. Channel or pore mediated involves substances that small molecules and ions that generally don't interact with their transporters. Carrier or transporter mediated substances have specific binding sites for the transported substance. This is passive, and active transport involves, that's why there's a little d and a big D right here. The active transport involves the transport of substances against a concentration gradient, so ATP is required. So that is the end of our Chapter 9 lecture. Thank you for your attention, and I will see you in class.